Hello, everyone, and welcome to today's webinar, Exploring the Possibilities of Loop-Mediated Isothermal Amplification. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. I would now like to introduce to you our speaker for today's webinar, Gintarė Gervaitytė, Product Manager, Thermo Fisher Scientific, Life Sciences, and Laboratory Products Group. For a complete biography on our speaker, please visit the Presenter tab. Today's presentation is interactive, and we have several polls for you throughout the presentation. You may also submit any questions that you have by simply type them into the Ask a Question box and click Submit. Your questions today and those submitted during the on-demand period will be answered via email at the address you provided at the time of registration. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Welcome, Gintarė. You may now begin your presentation.
Thanks for this nice introduction, and hello to everyone that are interested in loop-mediated isothermal amplification technology. Today, our topic is exploring the possibilities of loop-mediated isothermal amplification. Today, we will present the LAMP technology itself, the trends related to LAMP technology, its advantages, and what to consider about LAMP technology. We will also talk about applications and the ability of custom commercial supply. So let's start with the introduction to LAMP technology. In the face of the rising prevalence of infectious diseases, the need for rapid testing has never been more important. Swift and accurate diagnosis is crucial to curb the spread and impact of these illnesses. Technologies like loop-mediated isothermal amplification can be a great solution for fast and simple detection of infectious diseases. Because of the method's simplicity and rapidity, it has become a valuable technology for on-the-spot diagnostics and can be adapted to create molecular tests suitable for point-of-care applications. So what is LAMP? LAMP is a method for fast and simple DNA amplification under isothermal conditions that utilizes a DNA polymerase with strong strand displacement activity, such as BST DNA polymerase. One of the most popular modifications of LAMP is RT-LAMP, which incorporates reverse transcriptase to create a cDNA template from RNA. Now I will explain more about the principle of this method. In the context of LAMP technology, the principle of strand displacement is key to the amplification process. Strand displacement begins with a primer binding to the target DNA sequence. Then DNA polymerase enzyme, such as BST DNA polymerase, which possesses strand displacement activity, synthesizes a new strand complementary to the target sequence, and double-stranded DNA will form. Strand displacement will continue again on the new double-stranded DNA. As this new strand is synthesized, it displaces one of the original template strands. The displaced strand serves as a template for further amplification. DNA polymerases with strand displacement activity are essential to LAMP technology as well as to most other isothermal techniques. Here in this table, you can see the most common strand displacing polymerases that are suitable for different isothermal amplification technologies. The differences among strand displacing polymerases arise from their origins, characteristics, and specialized functions. Each polymerase type has unique advantages that align with specific applications. In terms of LAMP, BST and BSM DNA polymerases are the best choice because they are thermostable and can work at high temperatures, usually from 60 to 65 degrees, which are required for the LAMP method. However, BST DNA polymerase shows better performance, including speed and sensitivity, compared to BSM DNA polymerase. In terms of other polymerases, such as Phi29 DNA polymerase, this polymerase is often used for rolling circle amplification and whole genome amplification because of its ability to amplify long DNA fragments with high fidelity. Now, let's look deeper into LAMP method and how does it work. LAMP amplification occurs in two steps, non-cyclic and autocyclic. The first step is primer extension from the inner primer, FIP, followed by strand invasion extension from the outer primer, F3. The converted inner sequence is released and a stem loop structure is formed at the F-linked end. The same process is repeated on the other end with BIP and B3 primers, resulting in a dumbbell structure with stem loops on both ends. The structure contains multiple sites for amplification initiation and facilitates DNA amplification by autocycling, resulting in multiple lengths and cauliflower-like structures of amplified DNA. I will now pass it to Christine for our poll questions.
Thank you, Gintarė. This brings us to our first poll question. Which application of LAMP are you using or planning to use? Please select one of the answers. A, pathogen detection. B, genetic testing. C, environmental monitoring. Or D, other. And if you choose other, we'll give you an opportunity to specify on the next slide. Again, the question is, which application of LAMP are you using or planning to use? A, pathogen detection. B, genetic testing. C, environmental monitoring. Or D, other. Moving on to the next slide. If you did choose other, please specify. Just type your answer in the blue section that says enter your answer here. And we'll give you just a moment to type in your answer. Again, if you chose other, we'd love for you to specify. Thank you for your participation. Gintarė, back over to you.
Okay, thank you. After diving into the details of the LAMP method, it's time to shift the focus to the existing trends. Let's start from the beginning. The LAMP method was developed in 2000 by Notomi and colleagues, and from then, the method's popularity has been on a consistent rise. As you can see from Google Scholar results, the method's growth experienced a notable jump in the period of 2019 to 2021 because of the COVID-19 pandemic and expired patents for LAMP technology. Because of LAMP's simplicity and high speed, the method has emerged a popular technique for SARS-CoV-2 detection. According to Google Trends data, the frequency of the search terms LAMP and RT-LAMP also increased during the pandemic, which showed the method's popularity. Summing up the collected results, the key factors driving the growth of the LAMP segment in 2019 to 2021 were the increasing demand for rapid and highly sensitive test kits, growing cases of SARS-CoV-2, and growing need for isothermal nucleic acid amplification technology tests and systems for SARS-CoV-2. Currently, lamp and other isothermal amplification techniques remain interesting for researchers because of increasing funding initiatives for molecular diagnostics and infectious disease diagnostics, as well as the high prevalence of infectious diseases. Now I will pass it to Christine again for our poll.
Thank you, Gintarė. Our next question, would you like to get a quote for Thermo Fisher Scientific's LAMP products, such as BST DNA polymerase, superscript 4 RT LAMP master mix, or BSM DNA polymerase? Please select yes or no. We'll give you just a moment to answer. Again, the question is, would you like to get a quote for Thermo Fisher Scientific LAMP products, such as BST DNA polymerase, superscript 4 RT LAMP master mix, or BSM DNA polymerase? Please select yes or no. Thank you for your participation. Gintarė, back to you.
Okay, so now let's move to the advantages of LAMP technology. There are 4 main advantages of LAMP. The first one is that the LAMP method doesn't require expensive instruments like thermal cyclers because DNA amplification is performed at a constant temperature. LAMP's isothermal conditions and minimal equipment requirements make it suitable for point-of-care testing in resource-limited or remote settings where traditional PCR setups may not be feasible. Also, the LAMP protocol is quite fast. It takes less than 30 minutes to perform a reaction. This speed is valuable for diagnostics and time-sensitive research. Furthermore, this method has multiple detection options, including real-time, endpoint, lateral flow, and other detection methods. The LAMP method is also quite simple. Only a few steps are required from the sample to result. Additionally, the interpretation of results is quite easy. The products of LAMP can be observed even with the naked eye immediately after the reaction ends or even while the reaction is running. After talking about LAMP advantages, it's also important to consider some factors that can impact its application. There are few things that you need to consider when you choose LAMP technology, including primer design, carryover contamination and false positive results, non-specific amplification, and compatibility with downstream applications. The first thing to consider is primer design. LAMP reactions result in high specificity because of the use of specifically designed primers. LAMP employs from 4 to 6 primers binding 6 distinct regions of target DNA. It includes FIP, BIP, and F3, B3 primers and 2 loop primers. LAMP primer design is similar to PCR, but significant differences exist between the two approaches. LAMP primers entail several critical factors such as primer and amplicon length, melting temperature, GC content, secondary structure, stability at the end of each primer, and distance between primers. Firstly, let's talk about the amplicon length and why it's important. Designing primers to produce a resultant amplicon size of less than 300 base pairs enables good sensitivity and reaction speed. In principle, amplicons exceeding 500 base pairs can be obtained too, but these long amplicons can result in poor sensitivity and long reaction time. The success of LAMP also depends on the relative distance between primers. The primers are designed in such a manner that the distance from the end of F2 to the end of B2 is between 120 to 160 base pairs. The primers are also designed such that the distance from the 5' end of F2 to the 5' end of F1 is between 40 and 60 bases. Another important factor is GC content. Ideally, primers should have GC content between 40 to 65% with an optimal value of 50 to 60%. It is important, particularly for inner primers, that primers form no secondary structures. Additionally, it is important to avoid single nucleotide or dinucleotide repeats. The next aspect that should be considered is primer stability to ensure good amplification. The 3' ends of F2B2 and F3B3 and the 5' ends of F1C-B1C should be designed in such a manner that the free energy is -4 kilocalories per mole or less. The 5' end of F1C after amplification corresponds to the 3' end of F1, so stability is important. Finally, the melting temperature of the primers should be similar across primer pairs. The difference should be less than 5 degrees. The recommended melting temperature for F1C, B1C is from 60 to 65 degrees, whereas the melting temperature for F2B2 and F3B3 is from 58 to 61 degrees. If the two melting temperatures are further apart, it won't be possible to find an annealing temperature that allows both primers to bind to the template DNA. Another significant thing to consider for reliable target detection is the potential for carryover contamination and false positive results. LAMP is a method that can generate vast amounts of DNA products, and there is a certain risk of carryover contamination that can cause false positive results. To avoid the contamination, we recommend to follow these guidelines in your laboratory. First of all, clean the laboratory workspace and all equipment thoroughly before and after setting up each experiment. Also, change gloves frequently after cleaning the laminar flow hood and prior to adding the NTC. When carrying out endpoint detection, make sure the NTC samples are handled before the positive samples and tubes are closed when positive samples are added. Try to prevent contamination of the stock reagents by aliquoting them into the working stock volumes. Furthermore, use aerosol-resistant filtered pipette tips and change pipette tips after each aspiration or dispensation. Use separate areas and dedicated equipment and supplies for DNA or RNA sample preparation and reaction setup and amplification. Discard used reagents if carryover contamination is suspected and replace them with fresh ones. Use uracil DNA glycosylase to control carryover contamination. In this method, the oxyuracil nucleosides are specifically incorporated into all LAMP reactions such that all amplified products contain uracil, making them chemically differentiated from target DNA. All subsequent LAMP reactions should be treated with uracil DNA glycosylase, an enzyme that specifically degrades uracil-containing DNA, leaving only natural target DNA intact for amplification and detection, and thereby effectively eliminating carryover contamination. The third important factor to consider is nonspecific amplification. The LAMP method is fast and robust, but can often result in nonspecific amplification, leading to false positive results. To help ensure reliable results and control nonspecific amplification, we recommend following these guidelines. Make sure that the work environment and reagents are clean. We recommend cleaning the workspace regularly to prevent contamination. Set up the reactions on ice to avoid nonspecific amplification products. To enhance specificity in LAMP, primer design and optimization are required. You can find more information about primer design at thermofisher.com. Also, ensure that the primer melting temperature is neither too high nor too low. Low temperature may result in a nonspecific primer binding, and high temperature may inhibit primer binding to the template. Other reaction parameters, such as reaction time or BST DNA polymerase concentration, can also be optimized. Depending on the primer design and template concentration, BST DNA polymerase amount per reaction can be decreased down to 1 unit. If endpoint detection is used, time can be an important factor for nonspecific amplification. Reducing the incubation time by several minutes may help distinguish the NTC from the sample. As I mentioned before, the LAMP method is robust and sensitive. However, the specificity of the reactions and the visualization of the results may be complicated due to primer dimer formation, nonspecific amplification, and cross-contamination when running a large number of samples simultaneously. One of the ways to increase the specificity of LAMP reaction and to improve visualization of results is by combining LAMP technology and CRISPR-Cas12 detection system. This method consists of two sequential reactions: amplification of RNA or DNA via RT-LAMP or LAMP, and detection of amplicons using a Cas12 endonuclease. Method relies on Cas12 endonuclease guide RNA complex that recognizes amplified DNA target sequences by complementarity, which activates Cas12 endonuclease, and it starts to cleave the non-target DNA or reported probe randomly. One example is described in the article developed by Zahir Ali and colleagues. In this article, researchers established a specific and sensitive detection system for SARS-CoV-2 and named the system iScan. They showed two different detection assays, one-step and two-step iScan. In two-step iScan, RT-LAMP reaction and Cas12 detection were performed one after another. Meanwhile, in one-step iScan, both reactions were performed in the same tube. In the picture below, you can see the principle of the method. First, they collected nasal swabs from patients and extracted total RNA. For the amplification of SARS-CoV-2 RNA, they used an RT-LAMP reaction. Amplified products were detected by adding Cas12 cRNA complex to the reaction together with a fluorophore quencher reporter. After the specific binding of cRNA to the target DNA amplicon, Cas12 nuclease performed collateral cleavage on the non-target reporter and degraded the probe. The fluorophore was then liberated, resulting in the production of a fluorescent signal. The positive fluorescent signal means that the RT-LAMP product is detected. For lateral flow detection, a lateral flow cleavage reporter was added to the reaction. Upon completion of the reaction, lateral flow readouts were performed by placing lateral flow dipsticks into the COS12 detection reaction, and results were observed after 5 minutes. To sum up, Zahir Ali and colleagues established a rapid, specific, accurate, and sensitive detection system that is suitable for large-scale in-field deployment for the early detection of SARS-CoV-2 carriers, allowing them to be effectively isolated and quarantined, thus limiting the spread of the virus. The last thing to consider while performing LAMP reactions is compatibility with downstream applications. While LAMP is a powerful and sensitive amplification method, it may not be suitable for all downstream applications due to the complexity of the amplified products, cauliflower structures of varying lengths. However, target amplification is so extensive that numerous modes of detection are possible. Here you can see detection methods that can be used together with LAMP. If your research requires amplicon quantification, we suggest using real-time detection by adding DNA binding dyes that allow to observe results in real time. If qualitative detection is enough, the best choice is to visualize DNA after the amplification reaction is complete by adding dyes for colorimetric detections or running your amplified sample on an agarose gel or utilizing lateral flow detection method. Since we discussed the principle of the LAMP method, its advantages, and what to consider in LAMP reaction, here I want to show you the main LAMP or RT-LAMP workflow that is used for pathogen detection. The first step is sample collection, usually swabs, blood, or other types of samples. Then, pathogen RNA or DNA should be purified and can be added to RT-LAMP or LAMP reaction. To perform the LAMP reaction, several components are needed, including BST DNA polymerase, primers, DNTPs, reaction buffer, and magnesium. For RT-LAMP, reverse transcription and RNase inhibitor should be added additionally. The reaction should be incubated at 65 degrees for less than 30 minutes. Amplified products can be detected by real-time detection or visualized on a gel, or LAMP products can be evaluated by the naked eye. In terms of gel electrophoresis, if the LAMP product is amplified, you will see the ladder-like DNA fragments on an agarose gel. In terms of evaluation by the naked eye, a positive outcome will be indicated by color change in the tube. Now I will pass the word again to Christine for our poll question.
Thank you, Gintarė. And now this brings us to our final poll question for our audience. Would you like to talk with the technical sales specialist team to help you make a purchase of Thermo Fisher Scientific LAMP products for your research? Please select yes or no. Again, to our audience, your final question is, would you like to talk with a technical sales specialist team to help you make a purchase of Thermo Fisher Scientific LAMP products for your research? Yes or no? Give you one more moment to answer. Okay, Gintarė, back over to you.
Now let's move to the LAMP applications. LAMP or RT-LAMP technology can be applied to a wide range of applications, including pathogen detection, molecular diagnostics research for food and water safety, and cancer research and diagnostics. Since the prevalence of infectious diseases is increasing, LAMP or RT-LAMP technology can help to quickly detect pathogens and diagnose diseases in early stages. Pathogens including viruses, bacteria, or fungi can be detected using LAMP or RT-LAMP. These pathogens can be collected from different hosts, such as humans, plants, or animals. If you look at the pie chart, the most common pathogens detected by RT-LAMP or LAMP, according to Google Scholar results, are SARS-CoV-2, influenza, and HIV. This distribution is quite logical because these viruses can change very quickly, spread easily between people through contact or body fluids, and affect many around the world. That's why rapid and sensitive detection methods such as LAMP are suitable for the detection of these diseases. In terms of molecular diagnostics research for food and water safety, the most common pathogens are Salmonella, Campylobacter, Escherichia coli, and others. LAMP technology is also used for the identification of meat sources in food, for example, by targeting species-specific mitochondrial DNA. It also could be used for the determination of plant species and for the identification of genetically modified organisms and food allergens. The last group of applications is cancer research and diagnostics. In this application, LAMP is used for the detection of metastasis, mutation markers, and analysis of DNA methylation. Now that we have explored the diverse applications of LAMP, let's turn our attention to the tools essential for the development of LAMP and RT-LAMP assays in the field of diagnostic research. Thermo Fisher Scientific provides several products for LAMP technology, including standalone engineered BST DNA polymerase that is glycerol-free, and also superscript 4 RT-LAMP master mix that has all required components for LAMP or RT-LAMP reaction in one tube that ensures better convenience. The master mix has the same engineered BST DNA polymerase, superscript 4 reverse transcriptase, and cytonine dye included in the package for real-time detection. Furthermore, I want to highlight the main features of both products. The reaction temperature range is wide, from 35 to 75 degrees, with the optimal temperature of 65 degrees, making the products adaptable for various applications. Both products ensure high tolerance to inhibitors, high reaction speed. They can amplify the product in less than 10 minutes, and also high sensitivity. So both products can detect down to 50 copies of the template. If you are interested in assembling LAMP reaction by yourself, Thermo Fisher Scientific can also provide other components required for LAMP or RT-LAMP, including Invitrogen RNase outer recombinant ribonuclease inhibitor, Invitrogen superscript 4 reverse transcriptase, DNTPs, and primers. If you want to learn more about LAMP and its products, we recommend visiting thermofisher.com/LAMP or reaching us after the webinar. After discussing LAMP applications and introducing the products, I would like to illustrate real experimental data and demonstrate the versatile use of the superscript 4 RT-LAMP master mix across different applications. In this slide, we demonstrate the use of Invitrogen superscript 4 RT-LAMP master mix for rapid real-time detection of genomic DNA from 4 different pathogens of humans, including Salmonella enterica, Staphylococcus aureus, Mycoplasma pneumonia, and human adenovirus. I just want to highlight that even though superscript 4 RT-LAMP master mix contains a reverse transcriptase to generate cDNA in RT-LAMP, it can be successfully applied to amplify bacterial or viral genomic DNA. As you can see from Figure 1, reaction speed and sensitivity were evaluated using serially diluted genomic DNA at concentrations of 500 to 2,600, 500 to 5,000, or 500 to 10,000 copies per reaction, depending on the pathogen being analyzed. The main conclusion is that real-time LAMP reactions incorporating superscript 4 RT-LAMP master mix detected all 4 gDNA targets in less than 20 minutes. These results demonstrate that superscript 4 RT-LAMP master mix can be used in the development of highly accurate, sensitive, and rapid LAMP assays for the detection of a broad range of pathogen DNA targets. If you want to read more about the experiment, we recommend reading the application note that is called Real-Time LAMP for the Detection of Human Pathogen DNA using superscript 4 RT-LAMP master mix. In this slide, we demonstrate the use of Invitrogen superscript 4 RT-LAMP master mix for real-time detection of RNA from human pathogens such as SARS-CoV-2, measles, and influenza. Figure 2 shows that real-time LAMP reactions incorporating superscript 4 RT-LAMP master mix detected influenza and measles RNA in less than 6 minutes, depending on chosen primers. In Figure 3, you can see SARS-CoV-2 detection results. Reaction speed and sensitivity were evaluated using different amounts of purified RNA from positive clinical samples. The results show that the superscript 4 RT-LAMP master mix could amplify SARS-CoV-2 RNA from positive clinical samples in less than 6 minutes and can amplify SARS-CoV-2 RNA faster than other available reagents on the market. Now I will present how superscript 4 RT-LAMP master mix can be utilized for plant pathogen detection. Here we demonstrate the use of Invitrogen superscript 4 RT-LAMP master mix for rapid isothermal amplification of genomic DNA from the widespread fungi Botrytis cinerea and Fusarium graminearum and Omycetes, Phytophthora coctorum, and Phytophthora plurivora. In this picture, you can see that the reaction speed and sensitivity were evaluated using serially diluted gDNA at concentrations of 500 to 5,000 and 1,000 to 5,000 copies per reaction, depending on the pathogen being analyzed. The main conclusion here is that the real-time fluorimetric LAMP reactions with superscript 4 RT-LAMP master mix detected gDNA from tested plant pathogens in less than 15 minutes. Furthermore, you can find details about the detection of the same pathogens using endpoint colorimetric detection in our application note, which is called Using Superscript 4 RT-LAMP master mix to detect DNA from fungal or fungus-like plant pathogens. Moving forward, we have another interesting experiment about the use of Invitrogen superscript 4 RT-LAMP master mix for rapid detection of potato spindle tuber viroid RNA extracted from infected tomato plants. Why it's important? So potato is one of the major food crops in the world, and viruses are the main threats to potato production through yield losses and degrading stalks of seed tubers. Apart from viruses, viral agents defined as viroids also cause serious problems in potatoes. Potato spindle tuber viroid, or PSTVD, is well known for its highly contagious nature, having a wide host range, and is readily transmitted by mechanical means. The experiment starts with the extraction of viral RNA from PSTVD-infected potato leaves. Then extracted RNA is amplified by our T-lamp using superscript 4 or T-lamp master mix. Amplified product can be detected with real-time detection by adding Invitrogen Cyto9 stain, visual evaluation of the color change induced by Invitrogen Cybergreen stain, and agarose gel electrophoresis with Invitrogen eGel precast gels. The main conclusion here is that with superscript 4 RT-lamp master mix, PSTVD RNA in tomato leaves is detected in less than 10 minutes, regardless of the total plant RNA input amount. Furthermore, if you're interested in a more detailed description of the experiment, we suggest reading the application note that is called RT-lamp of potato spindle tuber viroid RNA using superscript 4 RT-lamp master mix. Finally, a couple slides about addressing our customers' commercial needs. As the company, Thermo Fisher Scientific has a strong brand and value proposition, also a broad portfolio, and innovating technologies and world-class scientists. As a commercial partner, Thermo Fisher Scientific provides full customer support, from product selection and customization with R&D support to a robust supply chain of our materials and manufacturing scaling. At Thermo Fisher Scientific, contract manufacturing is conducted using the highest quality standards at ISO 13485 certified sites following regional regulations. Our technical capabilities and customer-focused account managers will help answer questions and guide you through the processes of diagnostic assay development, licensing, and intellectual property. Thank you all for being part of this webinar. Your participation and engagement are greatly appreciated. If you have any further questions or would like to discuss anything, feel free to reach out.
Thank you, Gintarė, for your time and your valuable information today. We would also like to thank our sponsor, Thermo Fisher Scientific, and Labroots for helping to bring this educational webcast to all of you. Before we go, I also want to thank our audience for joining us today. As a reminder, questions that were submitted today and those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and have a great day, everyone. Goodbye.